Welcome back to part 2 of the lecture on microbial diversity as part of the Environmental Biotechnology course. At the end of the last lecture, I left you with the idea that many factors lead to microbial diversity, and therefore it may be relevant to assess whether it can be defined on the basis of metabolism, ecology, and taxonomy. So let us look at the drivers of microbial diversity from these perspectives with a relevant environmental process. To explain the drivers of microbial diversity, we will take the example of acid mine drainage, as shown in this picture here. Surface mining operations often expose pyrite, an iron-sulfur mineral, to environmental oxygen and water. This triggers a slow chemical oxidation process that produces ferrous iron, Fe2+, and leads to the formation of an acidic environment. Under aerobic conditions, ferrous iron is rapidly oxidized to ferric iron by specialized microorganisms known as aerobic iron oxidizing bacteria. This microbial activity sustains a self-perpetuating reaction, releasing more ferrous iron from the mineral surface. As a result, these aerobic iron oxidizing bacteria play a critical role in driving this cycle. If we write out the half reactions for the electron donor and acceptor and calculate the Gibbs free energy, correcting for proton concentration, we find that the energy yield from this reaction is quite low, approximately 32 kilojoules per mole. Now, iron oxidation can also occur microbially at neutral pH. For example, near surface waters as shown in the picture. At neutral pH, due to solubility differences, we observe a stable equilibrium between ferrous and ferric iron. Interestingly, for the same redox reaction, the Gibbs free energy available at neutral pH is nearly 3 times higher than at acidic pH described in the last slide. This difference in energy availability has a direct impact on which types of microorganisms thrive under each condition. In essence, pH not only affects chemistry of dissolved ions, but also shapes microbial community structure by influencing the metabolic energy available to iron oxidizing organisms. To summarize this from a point of view of microbial diversity, while the metabolism in terms of the electron donor and electron acceptor remains the same under both conditions, the metabolic energy available to iron oxidizing organisms is vastly different. Organisms such as A. ferrooxidans have developed a metabolism that can survive under acidic conditions and perform iron oxidation, while organisms such as Leptothrix or Carasia do the same reaction but only in neutral pH conditions. On a phylogenetic basis too, these organisms are identified under different classes, with Leptothrix identified as a proteobacteria, while A. ferrooxidans are identified as acidobacteria. This alludes to the fact that the interaction of metabolism and ecology leads to a vast majority of microbial diversity, whereas phylogeny, on the other hand, is a measure of this microbial diversity. Here too, we can see that any metabolic trait such as nitrification, hydrogen oxidation, or dissimilative iron oxidation is not conserved in any class but appears throughout the tree of life, signifying that phylogenetic diversity is a representation of the interaction of metabolism and ecology. Therefore, we can summarize our learnings from this lecture to answer the questions on the factors driving microbial diversity. The answer to which is that primarily catabolism forms the basis of microbial diversity. Ecology is interdependent with metabolism, and we have now seen an example where microorganisms sharing similar metabolic traits can differ in their ecology. Finally, we see that taxonomy provides a measure for us to encapsulate microbial diversity, but it is not the cause of the diversity in the first place.